In this video, I will be talking about the structure of DNA and the components of DNA. I will also be making some comparisons to RNA. DNA, as you may know, is a double-stranded polymer which is often presented in this double helix form. A polymer is just a chain of monomers, and the monomer, in the case of DNA, is called a nucleotide, and all nucleotides consist of three components. First is the phosphate group. The second is the sugar, and the third is the base. So let's take a closer look at these components. First, starting with the sugar, all sugars in DNA are deoxyribose.
The carbons on the deoxyribose can be numbered.
1', 2', 3', 4', and 5'.
The 5' carbon is the carbon that is sticking up off of the ring, and as you can see, it is the only carbon that is not in the ring, so it is easily identifiable as the 5' carbon. This numbering will become important as we move forward to the structure of the nucleotide and the way that the nucleotides fit together.
The second component of the nucleotide is the base, which is attached to the sugar at the first prime carbon. Now, for DNA, there are 4 types of bases: adenine, guanine, cytosine, and thymine. And these 4 base types can be separated into 2 separate categories. The first being purines, which include adenine and guanine, and pyrimidines, which include cytosine and thymine. Note how purines always have 2 rings, whereas pyrimidines only have 1 ring.
Purines can be remembered by the mnemonic pure as gold, while pyrimidines can be remembered by the mnemonic cut pi.
And finally, the last component of the nucleotide is the phosphate group, which is attached to the sugar at the 5' carbon.
The phosphate group includes a phosphorus that is bound to 4 oxygens. Now that we've covered the basis of the nucleotide, let's take a closer look at how these nucleotides fit together to form a single strand of DNA. Here's a more realistic view of what a single strand of DNA looks like. And you can go ahead and identify the phosphate group, the sugar, and the base of each nucleotide. You can tell that the base is the base because it has nitrogens within the ring, and you can tell that the sugar is the sugar because there's an oxygen in the ring. And you can tell the phosphate is the phosphate because it has 4 oxygens coming off of the phosphorus. And I know that what I circled is just one nucleotide because the base is coming off of the first prime carbon of the sugar, and the phosphate group is coming off of the 5' carbon of the sugar. So I've gone ahead and color-coded the rest of the components of each of the nucleotides. And just a note that the phosphate group of the top nucleotide is cut off in this image. And here is a phosphodiester bond, which actually links the nucleotides to each other in a single strand of DNA. The phosphodiester bond is between the oxygen connected to the 3' carbon and the phosphate group of the next nucleotide. So now that we've gone through how a single strand of DNA is organized, let's see how 2 strands of DNA come together. The complementary strands are held together by hydrogen bonds between the bases of the nucleotides. These hydrogen bonds allow for DNA to be double-stranded, and double-stranded DNA has certain characteristics. So the first characteristic of DNA is that there are complementary base pairings. So adenine will always be paired to thymine, and guanine will always be paired to cytosine. And notice that a purine, which is guanine and adenine, will always be paired to a pyrimidine, which is cytosine and thymine. And the bases are going to be paired by
hydrogen bonds. And adenine and thymine have 2 hydrogen bonds, whereas guanine and cytosine have 3 hydrogen bonds.
The second characteristic of DNA is that the strands of DNA are going to be antiparallel to each other,
which means that they're going in opposite directions.
And as you can see, the nucleotides in this diagram are pointing in opposite directions of each other. So the DNA molecule on the right has its 5' carbons pointing towards the bottom of the screen, making its 5' end near the bottom of the screen as well. And the DNA molecule on the right also has its 3' carbons pointing towards the top of the screen, making its 3' end at the top. And on the left, you can see that this is the opposite direction. And you can think of antiparallel directionality like a road where the 2 sides of the street have opposite directions of traffic. And the third and final characteristic of DNA is that the phosphate and sugar groups of the nucleotides are going to form this backbone. And the backbone is going to be on the outside, while the bases are going to be on the inside of the 2 strands.
And this makes sense because the 2 strands of DNA are held together by the hydrogen bonds of the base pairings. Now let's look at the difference between RNA and DNA. First, RNA is often single-stranded, whereas DNA is often double-stranded. Now, there can be double-stranded RNA and single-stranded DNA, but the cell makes single-stranded RNA and double-stranded DNA. The second difference is in the sugar. You can see that in RNA, the sugar has a 2' OH group, whereas in DNA, the group off of the 2' carbon is hydrogen. And finally, RNA, as you can see, has a base type called uracil, which replaces the thymine that you see in DNA.